All right, class. So, we know that we have the cranial nerve too, or optic nerve here, and this spot is called the blind spot or the optic disc. All right. Few things. Lateral to the optic disc, around here, there is a depressed area, and this depressed area is called macula lutea. This is called macula lutea. This is the area which has only cones, no rods. So macula lutea has only cones, and this is the area of sharpest vision. Area of sharpest vision. Okay, that's macula lutea. All right. The eyeball can be divided into two chambers or two cavities. From lens to cornea, this is called anterior cavity. And from lens to all the way here, this is called the posterior cavity. The posterior cavity is filled with a jelly-like or jello-like fluid called vitreous humor. The vitreous humor is actually formed during our embryonic development. So the vitreous humor is jelly-like. The anterior cavity is filled with a very watery fluid, and that is called aqueous humor. It is actually produced by the ciliary body. Okay. Now we will focus on the anterior cavity. Okay. The anterior cavity, which is between the cornea and the lens, can be further subdivided into two chambers. Please hear me. From cornea to the iris, do you see this area? This is called anterior chamber. And from iris to lens, this is called posterior chamber. And remember, the anterior chamber and the posterior chamber together makes up the anterior cavity. So to review, lateral to the optic disc, there is a depressed region called macula lutea, which has only cones and no rods. This is the area of sharpest vision. The eyeball has two types of fluid. The anterior cavity, which is the space between the cornea and the lens, is filled with aqueous humor. The aqueous humor is very watery, and it is produced by the ciliary body. The posterior cavity, which runs from the lens to the back of the eye, is filled with a very jello-like fluid called vitreous humor, and the vitreous humor is produced during our embryonic development. Zooming into the anterior cavity, it can be divided into two chambers. The space between the cornea and the iris is called anterior chamber, and the space between the iris and the lens is called the posterior chamber. All right. So that concludes my whiteboard drawing of the eyeball. Now we will focus on the PowerPoint. We will quickly go over the PowerPoint and make sure we understand everything. Okay, and then at the very end of this lecture, we will talk about the visual pathway. And okay, so we will focus on the diagram on the right hand side. We see that the external most layer is the fibrous tunic, And the fibrous tunic has two parts. Posterior 5/6 part is sclera and anterior bulging 1/6 part is cornea. I want you to remember that cornea is transparent and avascular. However, sclera is the white of the eye made up of dense, irregular connective tissue. Both sclera and cornea do not have any blood supply. The middle layer, which is shown by the pink box here, is called vascular tunic. And the vascular tunic, as we mentioned before, has three components. Posteriorly, we have the choroid, which is highly vascularized structure. Anteriorly, we have the ciliary body. So I'll just do the ciliary body here. This is the ciliary body. Remember, the ciliary body is made up of two structures, ciliary muscles and ciliary processes. Contraction and relaxation of ciliary muscles help to change the curvature of the lens, resulting in flattening out of the lens and giving us a curved lens. And this is really important for accommodation of the eye. All right, very anteriorly, the vascular tunic, this part is called the iris, and iris forms the colored part of the eye. Okay, accommodation, and choroid has blood vessels. Okay, so choroid, iris, and ciliary body, all three regions of the vascular tunic has lots and lots of blood vessels. It's not only the choroid that has blood vessels, all right? And also we do see that from the ciliary body, we have the suspensory ligaments, which are holding the lens in place. All right. Moving on to the deepest layer, in yellow box, that's the retina. And if you look at the retina, it has an outer pigmented layer in a kind of tan color, and then a lighter color layer, which is the neural layer. Remember, the junction between the outer pigmented and the inner neural layer is called ora serrata, which is shown by this. Do you see this? That's the ora serrata that marks the boundary between the outer pigmented layer and the inner neural layer of the retina. If you look at the retina, it has, okay, so the retina is divided into outer pigmented, inner neural layer. We just talked about it. Looking into the neural layer of the retina, superficially, we have rods and cones. Know that rods are responsible for dim light vision, cones are responsible for color vision, and these are photoreceptor cells. Okay, deep to rods and cones, we have bipolar cells, which is the middle layer, and in this we have the bipolar cells, all these pink cells, these are the bipolar cells. These are the rods and cones. And then the deepest layer is the ganglion cells, right here. And if you notice the axons of these ganglion cells, these axons join together to form the cranial nerve 2, which is the optic nerve. In this picture, so these are the photoreceptor cells, rods and cones. These are the bipolar cells, and these are the ganglion cells. I want you to notice one thing. So outermost we have choroid, this layer which has a lot of blood vessels, as we see. Then do you see these tan colored cells like this layer? This is a pigmented layer, and then this whole thing is the neural layer. Now one thing you should know. Look at the amount of photoreceptor cells. Look at the amount of rods and cones. A lot, right? Look at the number of bipolar cells. Do you see less bipolar cells as compared to rods and cones? If you say yes, you are right. Now look at the number of ganglion cells. Do we see less number of ganglion cells as compared to the bipolar cells? Yes. I want you to understand that at each level, photoreceptor to bipolar to ganglion, at each level, there is addition of information. There is information convergence. So the rods and cones are stimulated by light, but they are communicating with the bipolar cells and converging the information. And similarly, there are many bipolar cells which are talking to only few ganglion cells, and at this level also there is neuronal convergence. Okay, so information is getting summed up between each layer of cells. That was my point. All right. Sorry, my bad. Okay. The posterior, at the very posterior side of the eyeball, where all the axons of the ganglion cells come together to form cranial nerve 2, that part is called the optic disc, also known as the blind spot. Why? Because there are no photoreceptor cells there, so no image formation. Lateral to the optic disc, there is a depressed area, as you see here in the diagram, and that's called fovea centralis. Within the fovea centralis, there is a space called macula lutea. I talked about macula lutea on the hand-drawn diagram. The macula lutea within the fovea centralis has the highest proportion of cones and almost no rods, and that is the area of sharpest vision. Looking at the visual pathway. So, light rays hit the photoreceptor cells and trigger the photoreceptor cells. Information then goes from the photoreceptor cells, goes to the bipolar cells. From bipolar cells, information goes to the ganglion cells. The axons of the ganglion cells come together to form the optic nerve. The optic nerve comes out of each eyeball and it crosses, as you see here. So these are the two cranial nerve, right? Optic nerve. The optic nerve crosses at this point. Do you see this point? This is called optic chiasm. And then it enters the brain as optic tract. Do you remember the definition of tract? Group of axons within the central nervous system. So the optic nerves crisscross at the optic chiasm and then it enters the brain as the optic tract. Then the axons, some axons go to superior colliculi of the mesencephalon, which are right here. So some axons come to the superior colliculi and some axons go to the green spot here, and that's the lateral geniculate nucleus of the thalamus, just no thalamus. And then from the thalamus, information goes to the primary visual cortex of the brain, of the occipital lobe. So to review, the photoreceptors stimulate the bipolar cells that synapse with the ganglion cells. The axons of the ganglion cells come together to form the optic nerve. The optic nerve leaves the eye and then cross each other at optic chiasm and then enters the brain as optic tract. Some axons from the optic tract go to the superior colliculi of the mesencephalon, and some axons project to the thalamus. From thalamus, ultimately, the visual information goes to the occipital lobe of the primary visual cortex of the brain in order to process that visual information. That's the end of eye lecture. I'll stop here and then move on to ear. Oops.